My name is Suresh Tyagi. I'm a endowed chair professor in the Department of Physiology, University of Louisville, Kentucky. And my research is mechanism of cardiac remodeling, and basically what we are looking in the lab that heart has number of cells, and like myocyte are very well studied, but the myocyte endothelial, how cell communicate at juxta interaction in the cell surface. So we are looking endothelial myocyte coupling. So in the past, people used to think that myocyte does everything, contraction, relaxation, but my lab is thinking out of the box. We're thinking that myocyte, at least relaxation or diastolic function, is endothelial dependent. Unlike in the previous, people did not think that myocyte function is also depend on endothelial. They think capillary endothelium is perfusing with oxygen and blood and bringing nutrition, but we think that endothelium also is contributing to myocyte or cardiac muscle function. That's one aspect. Second aspect, we are looking at homocysteine. Homocysteine is like a cholesterol of 21st century. And in this one, people took homocysteine off the radar because they did not see some effect of folic acid. But just take a break. If you see a pregnant woman, what clinicians prescribe a woman a folic acid. What folic acid does, it recycles back homocysteine to methionine. And if you have a gene get epigenetic modification with methylation. What it is, you get methyl group from methionine to put on DNA so it can control the promoter. But the byproduct of the methylation is unequivocally, you're going to make homocysteine. So during embryogenesis, when womb is developing, you have a lot of gene turn on, turn off by epigenetic modification of the DNA. But that modification generate homocysteine. So folic acid was recycling back to methionine. So we think homocysteine is very important in the lab. What we are thinking, if we have enough machinery to make that homocysteine back to a good product such as hydrogen sulfide. And hydrogen sulfide is another gas which can vasodilate, which can decrease your blood pressure, which can relax. So it is more protective gaseous neurotransmitter. And it is involved in epigenetics. And epigenetics also involved in gut microbiota, what we eat. And there's a lot of interest in what we eat, what we are. That's not like this. This is epigenetic changes. Like two twin brothers, one is obese, one is lean. But in one, there's a more methylation of the gene with gut microbiota. And in one, that methylation is inhibited. So DNA is same, but the epigenetic change with what we eat, and it modifies the DNA, can control the activity of the gene, and that one brother become obese, one stay lean. Same thing, two twins are hypertensive. One is more responsive to calcium channel blocker. One is more responsive to angiotensin II blocker. It's the same gene, so it's not all DNA, it's the epigenetic changes. And epigenetic changes unequivocally involve homocysteine. And that one aspect we are looking in the lab, how that homocysteine control the cardiac muscle function, and then by autophagic mechanism, which is autophagy is like a starvation. When you starve, you engulf some of your cells so that your lean weight is autophagy. Your fat cell dying when it's starvation. So it's a protective mechanism. Same time, it can be detrimental in some situation. So these, in a nutshell, those are the research we are doing in the lab.